Click for IB Biology. Here we're looking at viral structure, the diversity in that structure, and the sizes of these virus particles. You'll find a blank PDF of this presentation down in the description so that you can make your own notes as we go along. When complete, you can take the quiz to test your understanding of the content. If you want to pursue some of the finer detail, I've provided a prompt structure to assist you, and then there will be links to other videos at the end screen. So viruses are found either in what is known as the naked form or the enveloped form. The naked form refers to the idea that the surrounding capsule of protein known as a capsid encloses a nucleic acid. The enveloped viruses also have that structure. However, around their outside, they also have this so-called envelope, which is actually the membrane derived from the host cell in which they had replicated. And taking from that host cell, this lipid, phospholipid bilayer has within it glycoproteins, which are derived from the host, but the virus will often incorporate its own glycoproteins, and these are commonly referred to as spikes. The nucleic acid is either DNA or RNA, and ordinarily we put a small v in front to make it clear that this is viral. When examining inside the capsid, we find that there will be a single strand of DNA virus, and we call that ssDNA, excuse me. So that's a single strand DNA, and equally we would find the same thing with an RNA virus. So that would be an ssRNA. So we have this combination now that we could have a naked single strand DNA, or we could have an enveloped single strand DNA, and equally so single strand naked RNAs, or single strand RNA with envelope. So these permutations are all possible in the different viruses. Since single strand is possible, also is double strand. And then also we can see the designation of positive or negative, and this is referred to as the sense. So positive or negative sense, and this refers to whether, in the positive sense, this means that the proteins can be synthesized directly from that structure. So if we have positive messenger RNA, positive RNA, we can make the proteins directly from that on the ribosomes. Negative would mean that it would need to be transcribed into the alternative form. Let's take a look at the sizes of these structures. Small viruses such as the polio virus imaged here are around about 20 to 30 nanometers. Medium-sized ones such as influenza come in at around about 50 to 100 nanometers. And the large viruses around about 150 to 300 nanometers. And this particular one in this image here, this is HIV itself. There have in recent times been a discovery of what are being called giant viruses, and these range between 400 nanometers to 1500 nanometers. And all of these particular giant viruses so far that have been discovered have been parasites of the amoeba, the significance of which is not yet fully understood. So a tremendous range in virus structure based on enveloped or not enveloped, whether or not we are DNA, RNA, double strand, single strand, positive and negative senses, and then of course in the actual size of these structures. on the screen in front of you appearing are links to the lytic cycle, one of the life cycles of the virus, to the site, and also to the playlists where you can find more virus videos.